Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. So welcome to the third live session of Basics of Biology. And today we are going to discuss the problems what you have, or in case of you have any doubts, you can raise your queries and I'll be happy to answer. So I don't think you have any queries, but I thought I should discuss what we have learned so far in this particular course. So I'm going to share my PowerPoint slide, and very briefly we will discuss or we'll actually going to review what we have discussed. So probably that will good to initiate the discussion also. So do you see a PowerPoint, right? So in this PowerPoint, what we are going to do is we are going to learn, or we are going to actually review what we have discussed. Yeah, it's not showing up, right? Huh? Okay, so this particular course, we were having the 12 modules, and the overall objective was that we would like to illustrate the different properties of living organisms. So how you study the living organism, how the organisms are being developed or appeared on the earth, and then we went on to discuss about the even the molecular level and cellular level. What is this? All is about. Okay, so we started with the understanding about the classification of the organisms, and we discussed about the 5 kingdom classifications. We have discussed about the different types of kingdoms. So within that, we have discussed what are the different classes are present and all that, and then we have taken very systematically. We have taken the classification of the animal classification and the plant classifications. And then subsequent to this, once you understand the classifications and you understand the requirement of the classifications, you are also curious to know how the organisms are evolved and how they are actually being present on the current earth, right? So we also discuss about the mechanism of evolution. Then we also discuss about the different theories on how the life originated on the earth and other kinds of things. And we discuss about some of the crucial experiments like the Stenimiller experiments and the and Louis Pasteur experiments and so on. So these two are very important part of the basics of biology, because you have to understand the classifications, the reasoning behind why we are doing the classifications. Because why we are doing the classification? Because it is easy to understand. It is easy to study a particular organisms when you do the classification, because you cannot study all the organisms on the earth, but if you classify them based on some of the phenotypic features or other physiological features, then it is easy to understand the properties of a particular group. And evolution is also very important, because evolution is going to tell you how things are evolving and what is the reasoning and all that. So once we understand these two processes, we thought of understanding the cells. So within the cell, we discuss about the prokaryotic cell, we discuss about the eukaryotic cell, and then we also discuss about the properties of the prokaryotic cell, what is the cell wall, how it is made up of, and what is the difference between the cell wall in the gram-positive bacteria versus gram-negative bacteria, and so on. And then we also move on to the eukaryotic cell. We understood the differences between the prokaryotic cell and the eukaryotic cell. And then we understood the different organelles what are present in the eukaryotic cell. So we understand about the nucleus, the content of the nucleus, the anatomy of the nucleus, how the molecules are getting into the nucleus and how they are getting out, and what are different processes are happening within the nucleus. And then we also discuss about the other organelles like Golgi bodies, endoplasmic reticulum, mitochondria, chloroplast, and the plasma membrane. And once you understand the cell is made up of the different types of biomolecules. So we have 4 different biomolecules. We have the proteins, DNA, carbohydrate, and the lipids. And protein is made up of amino acids. So then we move on and we discuss about the biomolecules. So we discussed about the carbohydrate. We have discussed about the different types of carbohydrates. We have discussed about the carbohydrate metabolisms. And then we also discussed about the isomerism in the carbohydrate. What are the epimers? What are the other kinds of tautomerizations and other things present in the carbohydrates? Then we discuss about the lipid metabolism. We discuss about the different types of lipids, cholesterol, phospholipids, phosphosphingolipids, and all that. And then we discuss about the nucleic acid. So we discuss about the different types of nucleotides. So we have the purine nucleotides, we have pyrimidine nucleotides, and you have the deoxyribonucleic acid, you have the RNA, right? And then we also discuss about the differences between the DNA and RNA, and then we discuss how the DNA structures and all that. And then we all ultimately at the end we also discuss about the protein structures, protein functions, what are different types of structural organization present in the proteins. You have the primary structure, secondary structure, tertiary structure, and quaternary structure. And while we were discussing all these, we have tried to show you the couple of demos so that you can be able to use, you can be able to see or understand the different techniques what we use for the protein structure analysis. And subsequent to that, these biomolecules are actually the executory molecules which are governing the different types of activities within the cell. So we also understand about the cellular processes. So we discuss about the central dogma of life. So within central dogma life is starting from the DNA. DNA is giving the RNA, and RNA is giving the proteins. And all these are connected in such a way that they are relaying the message. So DNA is present within the nucleus, and it is producing the RNA, and then RNA is transporting out of the nucleus and present in the cytosol, and then it is producing the proteins. So the DNA is making its own copy by a process which is called as replications. Then DNA is making RNA with a process which is called as transcription, and the RNA is producing the protein with and the process is known as translations. So all these 3 processes we have understood in the prokaryotic as well as the eukaryotic cell, and how the different types of molecules are participating into these processes, how the replication is happening, how you can be able to study the replications, how you can actually be able to perform the same replication within under the in vitro systems, and you can be able to use that and produce the more copies of the same DNA with a process called polymerase chain reactions. And what are the applications of the polymerase chain reactions? Then we discuss about how you can be able to do the transcription. So different steps of transcription in the prokaryotic system and the eukaryotic system, then translation in the prokaryotic system and eukaryotic system. While we were discussing about the transcription, we have also discussed about the post-transcriptional modification as well as the post-translational modifications. Now, all these cellular processes are same within the cell, right? But the cells are getting evolved and making the different types of cells. And that's how you are actually going to have the different types of physiological processes. You have the digestive system, you have the nervous system, you have the circulatory system, you have the excretory system, and you have the respiratory system, and so many more, right? So all these processes we have discussed within the physiology, and at the end, we have also discussed it. If you see in the last module, we have also tried to summarize everything so that you can be able to review everything and revise everything just before the exams. So this is all about what we have discussed in this particular course. And as far as the exam is concerned, the exams are going to be the objective type. where you are going to have the 4 options, and out of these 4 options, one of the option is going to be correct. And you are going to have the 100 questions, and you are going to have the 3 hours to attempt these 100 questions. And there's no negative marking, and there is no if you have, so with this brief overview or the review of the course, if you have any questions, I'll be happy to answer. If you don't have any questions, probably we'll going to stop the live session. So I'm just waiting for your questions. So it seems, I think you all are very busy in preparing your lecture content, and you are preparing for the final exams, which is scheduled very soon, right? So there's no question from your side. So it is good that you with all the content and all the things are clear to you. So since there's no question, we would like to terminate, or we would like to conclude the live session. And I wish you all the best for your exam and SEM exam. And I hope that you all are going to do very well. Remember that the questions are going to be from the complete content. So don't be try to be go with the shortcuts that you will study only about the physiology or cellular processes or any such thing, because that is not going to work. Okay, so I have prepared the questions which are going to cover the whole content. So it's a good idea to go through with the assignment questions and go through the questions of the the whole content. Okay, so that it will be easy for you to to answer the questions, and it is easy for you to gain the more marks. So with this, I would like to conclude the today's live session, and I wish you all the best for your exams. Thank you.